A very good evening to one and all present here. It's Devdulal once again, and we are about to begin technical session 2 of this international webinar. The technical session 1 was a very lively and productive one, in which we got acquainted with cutting edge research of the respective speakers in their specific fields. We hope this fest of knowledge starts from where it was left today morning. For technical session 2, we have as speakers Dr. Giacomo Reina from CNRS Delegation Alsace, Strasbourg, Alsace, France, and Professor Ayusman from Pennsylvania State University, USA. We are extremely honored by your presence. I would like to request the organizing secretary for today's event and faculty member, Department of Chemistry, Assam University, Professor P.C. Paul, to address the gathering. So please.
Good afternoon, everybody. I welcome you all to the second session of our international webinar, Antiviral Drug Discovery. Creation of Armory for the Present and Future Pandemic, organized by the Department of Chemistry, Assam University, Silchar, India. This venture is in commemoration of the Silver Jubilee year of the establishment of Department of Chemistry at Assam University. There could have been no theme better suited than this against the backdrop of the present pandemic that has shaken the entire planet. 100 years back, a severe pandemic of the swine flu virus H1N1 threatened mankind in 1919. The 21st century, in particular, can be termed as the century of RNA viruses. The SARS-CoV-2 virus, or commonly known coronavirus, which has already infected more than 18 million worldwide, did not just appear unannounced. We have had strong hints of the outbreak of several RNA viruses from the same group or family in the form of SARS in 2002 that originated in China. The Middle East originated the MERS in 2012. Further, mutant species of viruses from the same family are expected to evolve with more contagious and resistant functions. Other forms of viruses like Ebola appeared from Western Africa in 2013. Prior to that, another form of virus, unlike the SARS group, known as HIV, became a threat to the population since 1983. To date, there is no permanent cure for HIV, though advanced antiretroviral drug can prolong life. There has been a proud scientific breakthrough in this century. While chemistry has the scope of generating new synthetic antiviral agents to provide answer to the understanding of the molecular level of targeting the proteins or receptor of viruses, biology explains the very anatomy and functions of the virus. It helps identify the possible targets available for attacking the viruses with the new synthetic agents. Both physics and computational sciences can provide information about the efficiencies of the new antiviral agents and also provide an insight into engineering the shape and size of agents for better drug discovery and stronger binding with receptor proteins or viral RNA. This webinar is therefore aimed at addressing the all too obvious rules for interdisciplinary sciences in assigning a research goal to target the coronavirus and also prepare for the pandemic of the future. Chemistry, physics, biology, and computational sciences must unite in this endeavor. We have already heard from two of our distinguished speakers in the morning session. Our first speaker from the University of Sydney, Australia, Dr. Aviva Levina, mentioned the application of biolayer interferometry, BLI, to the studies of transferrin cycle and to antiviral drug discovery. She has been part of a team that has pioneered the use of BLI to assist antiviral drug discovery via the rapid screening platforms for cyclic peptides. The outcome of her research is to apply BLI to study the conditions that lead to release of viral particle within the endosome, and to test the drug candidates that can prevent this process. Our second speaker from the South Asian University, New Delhi, India, Professor Ritu Gaur, is a virologist, and she has emphasized upon antiviral drug discovery, maturation inhibitors, a novel class of HIV antivirus. She has given us an insight into how viruses can be targeted with antiviral agents. Our second session is dedicated to the chemistry of nanomaterial that can be used for development of point-of-care diagnostic, carrier of therapeutic, and vaccine development. A number of research targets for the nanotechnology community are strongly recommended. They include blocking of viral replication and boosting the immunogenic response to the pathogen, among several others. Several biotechnology companies are scrambling to leverage nanotechnology aided by nanocarriers and nanoliposomes in developing a vaccine candidate for the novel coronavirus, relying on various approaches, all of which are being tested in clinical using the recombinant protein nanoparticle technology platform to generate antigens derived from the coronavirus spike proteins is another emerging technology in hand to enhance immune responses against COVID-19, which is close to market. This evening, we'll first hear from Dr. Giacomo Reina from the Institute of Cellular and Molecular Biology at the CNRS, Strasbourg, France. He will apprise us with the role of various nanomaterials as possible antiviral agents. Our last speaker is Professor Ayusman Sen. He is a professor of chemistry at Pennsylvania State University, USA. His specialties are nanomotor, catalysis, and new materials. I am privileged to welcome both Dr. Giacomo Reina and Professor Ayusman Sen to this session of the international webinar and thank them for accepting our invitation to share their valuable research output with our audience. They enable us to imagine new frontiers of the sciences of nanomaterials and their application in biological systems. I also welcome our participants and hope that they lend a passionate ear and take back significant scientific aspiration from these lectures. We also have an added privilege of having amongst us today the distinguished Professor Alberto Bianco from CNRS Strasbourg, France, as a special guest. Welcome to you, Professor Bianco. I request Dr. Devashish Sengupta of our department to moderate the session and introduce us to the next speaker. Thank you.
Thank you very much, Professor Paul. Hello, everyone, and welcome to the technical session 2 of the webinar on antiviral drug discovery, creation of an armory for the present and future pandemics. I'm Devashish Sengupta and will be moderating this session of the webinar. Albert Einstein believed, and I quote, "Imagination is more important than knowledge, for knowledge is limited, whereas imagination embraces the entire world." Stimulating process, progress, giving birth to evolution. Our presenters today share the outcomes of their imaginations with our large audience. There are two speakers in this session. Our first speaker is Dr. Giacomo Reina from the Institute of Cellular and Molecular Biology at the CNRS, Strasbourg, France. Dr. Giacomo Reina received his master's degree in chemical sciences and technologies in 2010 and his PhD in 2016 from the University of Roma, Tor Vergata. With a thesis on functionalization and applications of detonation nanodiamonds, he is currently a postdoctoral researcher in the group of Professor Alberto Bianco in the Institute of Cellular and Molecular Biology at the CNRS Strasbourg, France. He is a scientist belonging to the graphene flagship work, and he is responsible for material functionalization in solutions. In 2020, he was awarded with the JSPS scholarship for a 2-year research project in Japan concerning black phosphorus in cancer therapy. His research interests focus on functionalization and supramolecular interactions of carbon nanomaterials for imaging, therapeutic, and diagnostic applications. Without any further ado, I present to you our first speaker this afternoon, Dr. Giacomo Reina.
Good afternoon, everyone. Thank you for your kind introduction and for being here. And today I will give you a lecture about hard nanomaterial in time of viral pandemics. As they said, I'm a postdoc and I work mainly on functionalization of nanomaterials. And with this pandemic, it was really shocking for all of us. And we were working mainly on other kind of disease, but now we are starting new projects about antiviral materials. And for this reason, in the beginning, we started doing some bibliography, and we recently published a review about that. And today I will discuss and I will show you some recent results about hard nanomaterial against viruses and how they work, what are the strategies, especially from a material chemistry point of view, and then the new challenges. So here, so regarding the content, as I said, we will see how nanomaterial, after a brief introduction, can eventually stop the virus attack at the different stages, from the early stage, so blocking the entrance, then how block the infection when the virus is already inside of the host cells, and eventually how to work and to help the immune system to recognize the infections. So why should we address the virus research? It's pretty obvious now, but it should have been also before this pandemic, because if you can see from this chart, we had different kind of viral pandemics during the last 120 years. For instance, in 1918, we had the Spanish pandemic who killed 100,000 of people. Then we had in the late 50s, sorry, we had the Asian flu, which killed 1 million people. And then we had the HIV that is still ongoing. Now it's less dangerous, but people who get infected have to take drugs all of their life, and this drug has a cost, and so that not everybody can afford this. And then if you see after 2000, we had different kind of viral pandemic or pre-pandemic, such as avian flu in 2002, H1N1, the swine flu in 2009, MERS in 2012, Zika, Ebola, and now we are with SARS-CoV-2. So, of course, as they have already said, the scientists should work really together, should form clusters, especially from different areas, to try to solve this problem. So, in particular, in my case, we are trying to put hard nanomaterial in first side against the appearance of new kind of pandemics. So, what are nanomaterial? Hard nanomaterial has been already used since a long time inside of the biomedical field. We can see they have been used in drug delivery, gene delivery, and imaging. We can have multifunctional material where we can detect a disease, and we can also fight against this with the same scaffold. Then they have been used also in tissue engineering. Hard nanomaterial can be implanted or can be even used for cell differentiation. And also, more recently, they have been used also in immune therapy. So hard nanomaterial can help. In particular, this is particularly used for cancer therapy. They can help the immune system to recognize cancer. So to then help the immune system to heal and to kill selectively cancer cells. Hard nanomaterial in general have been used mainly, I have to say, in cancer, and the antiviral properties have been really less studied. But we think that now is the time where many people and many research groups are going to be interested in them. So, what are hard nanomaterial? This is the definition I gave. Non-polymeric, organic, or inorganic materials. So, as you can see, it's not the best definition. I have the really problem because this hard part of the nanomaterial is really difficult to define. Indeed, I can say it's non-polymeric, organic, or inorganic, but what you have to keep in mind is that it's a rigid structure. Generally, the material is linked, is a covalent material, and could be even ionic material. And then the size can be between 1 typically and 100 nanometers. So in this hard nanomaterial, we can have, depending on the kind of the shape, we can have zero-dimension nanomaterials such as nanoparticles, one-dimensions such as nanorods or nanotubes, and two-dimension nanomaterial such as nanoflakes. So why nanomaterial into the antiviral research? As you can see, let's start from the size. So, as I said, 1 to 100 nanometer, what does it mean? It means that we can have, and we have now the technology to build a material as small as a molecule of glucose, or as big, as you can see, as a virus. And the fact that the size is in this is so small and can be tuned will tell us also that this nanomaterial can interfere with the trafficking and can interact with cells in general, such as small molecules or big viral particle. Then the second thing is the physics. Indeed, we don't have to forget the physical part of this nanomaterial. For instance, they can be fluorescent material, and in this case, there are nanomaterial that we can consider unbleachable, meaning that we can always see fluorescence even after a long time of irradiation. Then the composition, here I brought the periodic table because really we can do nanoparticles of mainly all the elements present in the periodic table. However, of course, there are some who are much more interesting for antiviral. And then I'm an organic chemist as a base, so this is the part that we like most, the surface chemist. Indeed, we are able to not only prepare precisely this nanomaterial, but also to functionalize and to give to cover the surface of the nanomaterial with some functional groups that then can be useful. For instance, we can take a drug on top, a dispersant, an imaging, an antiviral. We will see it soon, but we can really play and tune the surface chemistry to obtain always more specific and more performing materials. So as I said, this talk will be divided in the three parts, considering the viral spread inside of the cell. So we will talk in first part about uptake inhibition, so how to kill the virus before the infection, then the interaction inside of the host cells, and then the immune stimulation. So let's go with the first. Let's start from antiviral surface. So we have seen with COVID and with this pandemic that it is really important. We have to wash our hands a lot of time. And a lot of research now is going also in this direction. So how can we build good antiviral surfaces that allow us, like from the door or something that everybody is going to touch, that is not then contaminated by the viruses. So let's start. This research has started from really early with salts. Indeed, it has been seen that copper, ionic copper in solution, is able to block influenza A, avian influenza, herpes simplex virus, and so on. So why this? Because copper is able to generate ROS in solution by its cycle between copper plus and copper 2 plus. And in particular, copper plus, if you can see, is able to demolish hydrogen peroxide, forming hydroxyl radicals that are extremely reactive and toxic for the virus. So in the beginning, they said, okay, let's, if we have a surface, we can, for instance, cover it with a copper layer and see what happens. As they have done in 2007, what they have seen with influenza A, so the swine flu. They've seen that there is a low surface area because the copper layer that they used was flat. There was a poor release of copper plus ions, and so generally a low antiviral activity. And this, of course, was clear because if we start from metallic copper, then after in contact with a solution, the copper will get oxidized, but then we have normally much more release of copper 2 ions instead of copper 1. And so we have a dramatically less antiviral activity. So as a consequence to this work, they have So as a consideration of this work, they have said, okay, let's try to use nano copper. So in this case, they use nanowire of copper, of metal copper. In this case, what has happened is that, of course, they were able to enhance the surface area, and so eventually to enhance the contact of the nanoparticle with the solution and the release of cations. But still, the most favored cations release was copper 2. So still we can enhance the antiviral activity of this surface, but we are not still happy with that. We can do something more. So the subsequent idea was to use, instead of copper, nanoparticles to change the precursor. And in this case, they used copper iodide. They made these nanoparticles directly from top-down approach, so just by grinding finely the copper iodide. And what they saw was that upon two kind of viruses, they were able to create a really high and efficient antiviral surface. And this can be due because the copper iodide is low soluble salt. So once it gets in contact with the solution, it doesn't dissolve. So we can apply it on different kind of surfaces. But then when we have the release of copper from the nanoparticle to the solution, the copper is already in its oxidation +1. So it's already in its active form. And then it's able, as you can see clearly from this graph on the right, is able to dramatically decrease up to 4 order of magnitude the presence of the virus on top of the solution. So then how this can be applied in our today pandemic? For instance, on the building of masks. Of course, masks of copper iodide can be a bit problematic because iodide still is something that can be toxic and also can pollute. So what they have tested in 2010, they are these kind of masks using copper nanoparticles, but in this case, copper oxide nanoparticle, which are that are less toxic. So as you can see, they took a mask and they impregnate 2% of copper oxide inside of this mask, in particular in 2 layers that are layer A and layer D. So then they did the test on this mask about the virucidal capacity. So as you can see from the filtering capacity, from the average side, compare the test mask to control, we don't have, we have mostly the same numbers. What does it mean? That these masks are not able to filter more efficiently. And in particular, also the mask that we are using now, these are masks with the European label, so the one that we are using in Europe, can efficiently reduce the concentration up to 4 order of magnitude of viruses. What was really nice from this mask was the virus survival. Indeed, after testing this mask with an aerosol of virus, they have been washed and they have counted the quantity of virus present on the mask, so the activity of the virus present on the mask. And what they have seen, as you can see from the table on the right, we have a considerable decrease of the virus contact. We are speaking about 3 order of magnitude less when the mask have been impregnated with copper oxide nanoparticles. And this is now a standard, and this kind of mask are now present in the market. So this is a powerful tool how nanotechnology can help us to go against the viral pandemics. So Going through, as I said, so sterilized surface or have an antiviral surface is nice, but we can also we may also need some drugs who inhibit the uptake of the virus inside of cells. And how these drugs works normally. Normally they have a high affinity onto the viral capsid, and so they trap the virus and so make it impossible to enter inside of the host cells. So one of these in this context, there are different kind of example. Here I give you this one with carbon nanodots done by Kwang et al. In 2019, so just last year. As you can see, what they have done is really simple, but really effective. So they have prepared carbon nanodots with benzoxazine monomer. In particular, this is a typical synthesis of carbon nanodots where you do an hydrothermal treatment. And then they have tested the ability of this nanomaterial to block viral entry. What is nice, they have tested on, as you can see, 5 kinds of viruses. And they have seen that there is a dramatic release of the infection of the cells compared to the control after interaction of the carbon nanodots, these carbon nanodots with these viruses. Why this? This has not really been well explained. What they have seen is that these carbon nanodots has a really high negative charge, and so can electrostatically bind the virus. This kind of viruses, both enveloped and non-enveloped virus. However, the mechanism, I cannot give you more information about the mechanism because it's still unknown. What you have to consider in this kind of publication is that this is a virostatic approach, meaning that the affinity of the virus and the nanomaterials, or even a drug, it depends really on the concentration. It's highly dependent on concentration. So as long as we don't dilute too much the solution, we can have this effect of absorption of the nanomaterial onto the virus. But as long as we start diluting the solution and then we inject it to cell, we can have then a restart of the infection. And this is because this method doesn't suppress the virus protein. It's just coated. So the virus is still active. It's just coated by the nanomaterial. So there are other ways how we can not only attack the virus, bind the virus, but also to make it harmless. For instance, again, from before nanotechnology, there was, let's say, solution technology. So it is well known that silver, for instance, silver and gold are a good antimicrobiotic system, in particular antivirals. And why? Because these two atoms these two metals, have a good affinity for sulfur. And for instance, in the case of silver and HIV, they are able to bind cysteine protein present on the capsid of the HIV, and so to inactivate and to block its activity. So unfortunately, the problem of silver is, again, its toxicity, because this action is not specific. So it is true that silver is already implied, for instance, in toothpaste or in some medical equipment to keep them sterile, but it's not, we cannot inject silver, or we can do it with really, really low concentration inside of our body, because it can be toxic. Additionally, this strategy has no targeting, as you can imagine, so we not target the silver ions where to go. We can just think about a topical administration, and there is no release control. So on the antiviral hard nanomaterials, maybe silver is the most studied, silver nanoparticles, because they are really easy to prepare. We can prepare different kind of particles with different kind of size, and we can really tune their shape and size. So why we prefer nanoparticles now? Because when we have a silver nanoparticle, we have the silver atoms in the core, and then we have the silver atom on the surface. These atoms have different properties that are really special. They have a high potential, and also the potential of release of these silver atoms is pretty slow, meaning that they are quite stable in solution, at least stable enough to have a constant release. And once we can reduce the release, the overall release, and instead we have a topic release close to the nanoparticles, what we get is instantly an elevation of toxicity. However, we don't have targeting with just silver nanoparticle, but we can moderate the release. And we can have a release control even if moderate, because it depends on size and shape. And the other problem of this particle is the low colloidal stability. You have to imagine that once these particles get in contact with cell culture media, they will immediately get in contact with proteins, start coronations, and other side effects that make them easily agglomerate and settle down. But their antiviral activity is really well known and studied. Here I show you an idea. In reality, the different kind of mechanisms that are now under study, because what the scientific community think is that there are different ways how silver is working. But in general, you can have the block of the virus both from the nanoparticle or from the released ions from them. So despite of this limitation that I told you, we can see that we have different the silver nanoparticle, pristine silver nanoparticle, have been applied on different kind of viruses, and as you can see, the active concentration is pretty low. We speak about microgram per ml, and they depending on the virus, as you can see, we have different kind of mechanism of action. So another way to get a better silver nanoparticle for as antiviral is, of course, use the surface functionalization. In this case, the nanoparticle can be easily, as I said, functionalized with different kind of group. And what does it do, the functionalization? Well, at first it helps with the release. Somehow the presence of a capping agent onto the silver surface will slow down the release. And since we can choose the degree of functionalization we want onto this surface, we can also tune the release. In addition, we have less formation of corona and of agglomerates. So again, we can, by this, we can have stable nanoparticles, reduce, of course, the toxicity for cells. And if we choose a good functionalization, a good capping agent, we can also have targeting possibilities. So functionalized silver nanoparticles have been also applied more recently into as antiviral materials. We can see that they have been applied again and against HIV, RSV, H1N1, and so on. And even with different capping agents such as fomic carbon, you can see from the first line PVP or BSA, so a protein, or they have been used with antiviral agent as capping agent, so eventually enhancing their activity as antiviral materials. So an application in real life. This I look till this morning, I still don't see any company who say that they use it, that they use silver for condoms, but still it has been published and it seems to work really well. So they have been implied inside of condoms. They have been tested for if they change the mechanical property of condoms, and it seems not. But what is more important is that when condom is coated with the silver nanoparticle, what they have seen is that as you can see from the graph on the right, they have a strong antiviral activity. So the virus in contact with this surface is deactivated almost totally. And more importantly, they have tested also if the solution of this in contact with this condom with silver is able to have an antiviral effect, meaning that there is a release of silver from the condom, and it seems not. So as you can see, the antiviral effect in this case is really modest. Meanwhile, in the case of non-coated polyurethane condom, we have our positive control, where we have a good, almost, let's say, absent antiviral activity. So another way of killing, how hard nanomaterial can kill, deactivate viruses. This is something really special. This is something we have never seen before with other, with classical, let's say, chemistry, with solution chemistry, and it's because they are hard, as I say. So this hardness makes the difference. Indeed, the nanomaterial can eventually interact with virus and can cut it down, as I did, as I show here in this GIF, in this image on the left. Indeed, it's in the beginning it has been theorized. For instance, graphene, it has been theorized that it can destroy Ebola viruses by intercalation. So it can be this sharp material can cut the virus capsid and so inactivate it. And this is driven by hydrophobic interaction mainly, but this is a theory, so it's a really starting point, because then we have problems, because graphene itself, it's really hydrophobic, so it's pretty hard to put it in solution. We need to exfoliate it, and then we would need eventually to test if this theory goes right, and I hope that this is on study now. What we know from other nanomaterial is that we can have this mechanical breaking by other mechanism, for instance, adsorption-driven breaking, where we have where it had been tested and see with graphene oxide with PDV virus. what they have seen is that first there is an interaction of the virus onto the surface of the graphene oxide, driven by a negative charge. And then this interaction, as they have seen, and we have seen already with other protein, graphene oxide can denaturate proteins onto its surface. So what happens is that we have a continuous denaturation of the virus that leads to the open of the capsid. And this has been proven also by TM, as you cryo TM, as you can see by the image on the bottom, where you can see a porcina endemic diarrhea virus, a virus that is intact, and after in contact with graphene oxide, you can see that there is a total loss of the capside structure. Wonderful. So another way, and this is really brilliant, to destroy mechanically viruses has been proposed by Professor Stellacci at the EPFL, and has been done again with the help of hard nanomaterials. So what he's a physicist, and he thought that if we functionalize a nanomaterial, hard nanomaterial with some ligands, as you can see here with the red dots, that are really are long enough and have a high affinity for the viruses, the virus protein will be like attached to this ligand, and the fact that our nanomaterial is hard will provoke a deformation of the virus, and so it's inactivation. And this was predicted and also true, as you can see here. From again the cryo-TEM, he did on 3 kinds of viruses using the same sulfonate ligand, showing that once the virus was in contact with these gold nanoparticles, but the gold is not the point. The point is that it's a hard nanomaterial, and with this long length, the virus was destroyed mechanically. So we have a mechanical destruction of the virus, and of course, its inactivation after. So this kind of mechanism of destroying virus is still really early. It's not really well studied. We have just few examples, but I think it's something really interesting because it's something, a plus that only nanomaterial, hard nanomaterials can give against viral attack. So now, another way is, let's say, to avoid that the virus enter into the host cells is to make think to the virus that our nanomaterial is the host cell. So in this case, we use cell mimic ligands. So that is a kind of example that I told you before with the gold nanoparticles. So what we have is that the nanomaterial is functionalized, covered with some cell mimicking ligands, and then the virus gets in contact with the nanomaterial and it's surrounded by the nanomaterial and is not able anymore to enter. So there are different kind of publication and examples of that. I will show you just I will show you this one because I find it really nice and a really well developed idea. So what they did was they used graphene oxide, they functionalized it with iron oxide nanoparticles capped with amines. So this these nanoparticles have been formed onto the surface of graphene oxide, and then the graphene oxide have been reduced, and then they play another radical chemistry to attach onto the surface of graphene oxide the sulfoxide acid. So why? So in this case, they use a magnetic probe that is iron oxide. So to eventually discard and separate the nanomaterial after the treatment for the solution, so you can recycle it. And then you use this sulfonic acid, of course, the salts, to mimic the cell, so to attach to help the binding of the herpes simplex, in this case virus, inside of the onto the nanomaterial. Moreover, they reduce graphene oxide in order to have a good photothermal therapy, meaning that upon irradiation, this nanomaterial is able to heat, to generate local heat that will denaturate and remove the viral activity. And so all these complex process against herpes simplex virus, they have seen that the relative infection efficiency without near infrared is the same. So this system work only when we apply a laser. So when we use a photothermal therapy. What does it say this to us? As I told you before, we not only have to bind the virus, we have also to inactivate it, because as long as then we start washing our system, the dilution will let the virus, active virus, into the solution. And so we will continue to have it active and infective. So as I said, this virostatic it can be a problem because we need to have enough concentrated nanomaterial to see the effect. I'm really sorry. I had some problems with the presentation, so I cannot show you here any animation, but there is one huge pro that we have when we use nanomaterial, that is multivalency. Basically, when we consider the binding, the thermodynamic of the binding between a receptor and a protein and a ligand, this is it has of course a constant of affinity, and it's ruled by the concentration. But if we have more ligands inside of the same molecule, you have to imagine that the local concentration of the ligand close to the active site is really high. So we will have the possibility to bind more, and we will be able to enhance the constant of the binding constant of this approach. All of this to say that once we get highly functionalized material, the more they are functionalized, the more they will be effective. And so we will be able to really reduce their active concentration significantly. One good example has been done by fullerene. Fullerene maybe are the second most studied nanomaterial for antiviral activity, in particular for cell mimicking. In this case, fullerene, they don't mimic the cell, we can say, they mimic the virus. So the fullerene has been used, functionalized with mannose. Mannose is highly recognized by a receptor, lectin, present on cell, and lectin is a receptor that is targeted by different kind of viruses. So the main idea was, okay, if we saturate the lectin expressor onto cells, we will avoid the virus can enter into cells. And for this reason, there is a huge chemistry that I won't go into that, because there are different kinds of reactions and strategy, but what you have to see in the big scenario is that they did these sugar balls, meaning that they functionalized this ball of fullerene with sugars, with mannose, and they were able to do direct functionalization onto the surface of the fullerene. They have changed the spacer. They have done this other sugar ball structure with one fullerene in the center and then all the other functionalized. Or they have also used the supramolecular interaction fullerene, creating this kind of micelles of fullerene with mannose. So, as I said, there are different kind of example about that, and there is a lot of organic chemistry onto that. But what I want to show you is that this strategy has been applied to different kind of viruses, in particular Ebola and dengue, and there is something also on Zika. And as I told you, this multivalency effect has a huge importance. Indeed, if you can see the last examples, for instance, you can see against Ebola with this liposomes make of fullerene, the EC50 was only 424 nanomolar, meaning that if we multiply this by 6, is like more than 2000 nanomolar of sugar to block the viral infection. This is impossible without nanotechnology, because even if we go up to molar concentration of sugar, we are not able to block the viral infection, and this is due and really strengthened by the multivalency effect. So let's start to the stage 2. So how hard nanomaterials can work as antiviral once the infection has started, so inside of the cells. As I said, nanomaterials are already well known and well used for drug delivery, so they're able to ship to different kind of cargo molecules, such as drugs and antiviral agents. So here there are some examples in the literature. Here I show you a really simple one where they use the graphene oxide. Here they call NGO, and they use an DNA enzyme specific for hepatitis virus. So what happened here is that when the enzyme is alone, is not able to enter inside of the cells, but when it's in the presence of graphene oxide, the enzyme can be delivered inside of the cell. And more importantly, they use for this fluorescent marked DNA enzyme. And they see that there is then a release of this DNA enzyme inside of the cell only when the infection is going, meaning that the presence of viral DNA help the release of the drug, in this case this enzyme, and then they have seen that there is quite fine antiviral activity upon the use of graphene oxide loaded with this enzyme. So to make the long story short, they have seen they have created an antiviral material, and they have created also a sensor, because these hepatic cells, once they are infected and treated with this nanomaterial, become fluorescent, so we can see the infection spreading. So another way this has been done in vitro. Another nanomaterial used for drug delivery has been carbon nanotubes. In this case, they have been used for grass carp and for grass carp rhinovirus. And in particular, in this study, they have oxidized first carbon nanotubes. Then they have functionalized them with BSA, and then they have used ribavirin, that is an antiviral agent, functionalized to BSA via a ester formation. They use this ester formation because it can be cleaved inside of the biological fluids, so we can have the release eventually of the ribavirin. And they have seen in vivo that there was not evident toxicity, and that the antiviral activity was particularly higher in the presence of carbon nanotubes compared to ribavirin alone. And also, if you can see from the right, they have seen that there is also a higher release of ribavirin. This because the carbon nanotube can penetrate better the cell barriers and tissues, so we can see that the ribavirin is more diffused and reach a higher peak in the more or less the same time scale. So overall, they have seen that this system was working better without side effect. So another example, again, fullerene, as I said, they are really well being studied, is their activity with HIV. This starts from a paper in 1993, where they were theorizing that an enzyme of HIV, the protease from HIV, could be inactivated by fullerene, because the pocket of active site of this enzyme is perfectly filled with the fullerene. So this was something really nice, but really difficult to to prove, because fullerene is totally hydrophobic. So we need to vehicle with DMSO or other solvents. So in this, starting from these results, we had different kind of researchers work on that, and in particular working on drug screening. So doing different kinds of modelization to see if there were some organic groups onto the fullerene that would help to enhance their inhibition activity to HIV, and also enhance the solubility. And I show you just these two papers. I don't know if you are able to see the images, but what I want to tell you is that upon functionalization, we can sensibly reduce the EC50 with a good C50, so meaning that we are able to kill or to inhibit the virus spread without generate high toxicity. And in this case, we are in the submolar sub-micromolar, sorry, scales. So this is another strategy. Unfortunately, all of these studies up to now are in vitro. So we still don't have information about the in vivo applicability of these systems. So here you can see this. This was a picture we have prepared. So how fullerene work is membranotropism, so its ability to enter inside of cells, and the different kind of functionalization they have applied to against HIV. So another way that we can find, and that this is something really also nice for drug delivery field, is also to use some particular activity of our nanomaterial. We have seen that fullerene via functionalization can be used as a drug per se, And here we can also not only reduce the infection, but we can also reduce the side effect of the infection, for instance, cell viability. So we can somehow slow down the virus, but also help the cells to survive and so to have less damages from the infection. And one case study, the case study that I propose you is with selenium nanoparticles. The selenium nanoparticle, selenium is a ROS scavenger, and during the virus battling and when the virus starts to infect the host cells, we have an enhancement of ROS, basically of the radical stress inside of the cell that may lead to cell death. So in this case, they used selenium nanoparticles because they are able to penetrate easily the cell membranes and they can reduce ROS with ribavirin, that is again an antiviral agent. We can see that compared to mock in vitro, we have a higher viability when infected cells are treated with selenium nanoparticle with ribavirin compared to the use of the two, meaning that the ROS alleviation plus the antiviral effect of ribavirin are working together. And as you can see, they have done Western blotting, monitor the caspase-3, that is an indicator of apoptosis, so saying that if the cell is dying upon ROS stress, and they have seen that there is a general reduction of the caspase-3 activity, indicating that the cells are reacting well, and the ROS stress is relieved. But more importantly, they have done a preliminary study in vivo. Here you can see the slices of the mice lung. And as you can see, with the mock, we have the formation of infections inside of the pulmonary regions. Meanwhile, when we use ribavirin, we have a bit less of this. And with the selenium nanoparticle and ribavirin, we can see that the pulmonary the pulmon looks really similar to the control. And so indicating that most probably we are having a release of the radical stress. So as you have seen, I have talked with you a lot about radicals. We have seen the copper in the beginning for the antiviral surface, and then here we have seen the selenium. But as you can imagine, the radical stress is really important and something that has been studied for antivirals. And in particular, is important for our next topic, that is the stimulation of innate immune system. Indeed, this radical stress produced by the virus infection is able to stimulate gene producing interferons. The interferon is alpha in particular, is a molecule that promote inflammation and alert the immune system that an inflammation is happening, and also block the viral replication inside of the cell. So, as I said, if we reduce the ROS, we can enhance the survival, but eventually we are not able to alert the immune system. So another way instead is to create interferon, and so to create an inflammation, so to give a signal to the immune system that a viral invasion is happening. One One example has been done by the preparation of a composite of graphene oxide, again as a deliver, with silver nanoparticles that we have seen have really good antiviral activity. So as I told you, we have different kind of mechanism. The graphene oxide with silver nanoparticle is able to block the entry, but also to stimulate the interferon synthesis gene, and so stimulate the inflammation. And they have seen it with two different viruses. They have seen that this nanomaterial has a good antiviral activity. Here you can see from this immunostaining. So in green you can see the infected cell, and when we use when the cell have been incubated with graphene oxide with silver nanoparticles, we have much less green, indicating that we have much less viral proteins. But also via ELISA, they were able to measure the production of interferon alpha and other pro-inflammation cytokines, and they have seen that the inflammation is much higher with the nanomaterial, and so the spreading of the cell-to-cell spreading of the virus is highly reduced. So again, this slide maybe with animation would have been better, but let's try to follow me. Let's start from the beginning. So let's see now the adaptive immune response. So in general, what happens is that the antigen, in this case the virus and the virus protein, when they enter in contact inside of the body, they get in contact with APC, that are antigen proposing cells. In this case, I draw the dendritic cells. Then the virus get internalized, and these cells are presenting the the viral protein to other sentinel cells. And after that, this stimulated immune system to produce a specific antibody and to have an adaptive immune response. So when we go to vaccination, what we are doing is to try to stimulate an artificial immune response through through viral parts. Let's say normally we use the viral protein. We want or inactivated virus. We want an artificial response, because we would like to be immunized, of course, but without without the virus, so without get infected. And sometimes what happens is that this classical vaccine doesn't work always, and because this protein or this part of the virus that we use are not able to generate an appropriate immune response. And for this reason, they are called, they have a low immunogenicity. So it has been seen and studied and used also with soft nanomaterial, that when we take the viral protein and we attach it to something that resembles the virus itself. For instance, here hard nanomaterial, we can prepare virus-like particles. This virus-like particle can be much easily recognized by the immune system to generate an immune response. And in this case, we speak about nanovaccines. So in this case, the nanomaterial in general is used as a carrier to propose the antigenic molecules to the immune system. So one example has been done by zinc. Zinc, I didn't had the time, I didn't told you before, but it's like copper, so it's able to generate ROS upon immersion in solution. Due to its cation. So zinc has been used in this case, zinc oxide teardrops. You can see here this nice nanomaterial with a really nice morphology has been used for the treatment of herpes virus in vivo. So what they have seen, what they have done is incubating the zinc nanoparticle with the virus and then used photodynamic therapy in this case. So they have irradiated the zinc in order to generate more ROS and and kill more viruses. So then the virus stay attached onto the zinc surface. And upon of that, they have then given to mice, and mice have been challenged with the herpes virus. As you can see, in the mock case, we have a mortality that is 100% in the first 5 days, while when the mice have been treated with zinc oxide teardrop nanoparticle, we have a totally recover of the viability. So the virus is not able to kill these mice. And here you can see they have monitored the lesions that happen after the first 5 days of the challenging. So as you can see with in the case of the mock, the vaginas of the mice presents different kind of lesions. Meanwhile, with zinc oxide nanoparticles treatment, we have that the mice seems with no lesions. And more importantly, to see if this treatment was able also to generate an immunization, they have taken the splenocytes, where there are TMB memory cells, and they have incubated them with infected cells to see if they were able to recognize the viruses. And as you can see from the control, we have this is a fax. You can see the death cells, meaning that the cells have been recognized and killed by the immune system. You can see that upon use of the nanomaterial and irradiation. So when the nanomaterial has been used and has been used also to propose with the virus, we have a higher presence of dead cells, indicating that this nanomaterial not only has prevented the viral attack, but also has helped the immune system. And now the mice that have been treated with zinc oxide nano teardrop have been immunized. So last example that I will show you is with using calcium phosphate nanoparticles as a nanomaterial. Here, what was really interesting from my side was not only the use of this hard nanomaterial. Why this one? Because it's really biocompatible and biodegradable. The chemistry that they have done, but also because they have used reversed vaccinology against herpes virus. So they have used 2 different kind of peptides that have been first screened by the reverse vaccinology, and they have seen that in general we have the peptide alone doesn't have a good immunogenic response. As you can see here from this graph, we cannot really reduce the infection. Meanwhile, both the peptide works really well in the case of immunization when they are linked to the nanoparticles. Indeed, you can see that the number of infected cells upon 3 number of vaccination is really low. It's practically 0, and this has this has also an application with the survival rate. Indeed, we can see that when we use the vaccine, both with the MOC or with the 2 peptide P1 and P2 alone, we are really not able to immunize the mice, and the mice die as with the infection alone. Meanwhile, when we use the calcium phosphate to deliver these antigens, we can see that especially in the peptide 2, we have 100% of viability of the mice used, indicating that we were able to generate a strong and good immune response. So what are the conclusion and future challenges? We have seen in this presentation that hard nanomaterial can be used for surface disinfection, can be used to bind and inactivate viruses prior the entrance, and can deliver antiviral agent and tune the immune response. We have seen how it works with the adaptive and innate immune response. What are the future challenges? So when we started to write and to work on antiviral, we have seen that there is a really publication funnel. Indeed, we have a huge number of publications speaking about the surface sterilization, uptake inhibition, but as long as we move to in vivo tests, we have always less and less. And I think we need to put our effort to start new collaboration and to work with virologists and biologists in general who can test and eventually give us a new idea on the formulation of new kind of antiviral agents. As in general, we still have not seen that much the implementation of other physical properties of the nanomaterial, such as photothermal and photodynamic therapy. And you have seen, I just show you few examples about the mechanical braking, which I think that can be an interesting topic. As I said, we need, there is a lack on in vivo testing, there is a lack of targeting. We haven't seen such high chemistry except for some cases such as fullerene. In general, we can still, the chemistry that has been developed onto nanomaterial has not been totally used. And there is also a scarce information about allergy that this kind of nanomaterial can give, and also that is something that is really slowing down the application of hard nanomaterials in clinic. And in this context, I would like to say that I work, as I said, in the graphene flagship. We are working mostly on graphene and graphene family, and we are trying to speak up and to really link ourselves onto the standardization of the material preparation, standardization of the functionalization, because else all these papers seems really dispersive and seems really not useful, because it's really hard then to compare two different results. There are still, in this way, we have to work still a lot. We are acknowledged that it's much more complicated, because nanomaterial can have different parameters that are important, such as size, shape, and surface, but still it's something we need to do if we want to go in vivo one day, or as has been already done for gold nanoparticle, iron nanoparticle, and some other few nanoparticles that have been clinically tested and approved. Then we have to do a general assessment of toxicity, this for sure, and also study long-term toxicity, the implication that they would have for the environment. And in this context, the lab of Dr. Alberto Bianco is one of the maximum experts in biodegradation of these nanomaterials, so to see if they are biodegradable, and eventually if we can avoid to have bioaccumulation inside of the body. And are we working on antiviral nanomaterial? Yes, we have started really recently during the confinement. I was lucky enough to start this project with my boss. Here you can see Dr. Alberto Bianco and Dr. Paolo Samori. We're starting with working with graphene materials for antiviral activity. We start a new collaboration with a virologic group in Switzerland. So we are really happily starting this new research area. And with this, I would like to thank you all for your attention. And all these results and much more has been published recently on ACS Nano. And we are really happy we have been selected as COVID and in general antiviral paper. So you can download it for free for 1 year if you are interested in more. And regarding myself, here there are my contacts. As they told you, I'll be in Japan really soon, so I won't have any more my first email, so I will give you my personal one, and you can always contact my boss, Dr. Alberto Bianco, that would be really happy to share a project and interest with you. Thank you a lot.
Thank you very much. That was a brilliant rendition on the review of hard nanomaterials that have been recently used as antimicrobial agents, potentially being next generation drugs to fight viral infections. Hard nanomaterials, or HNMS, can block infection in early and middle stages and are also able to mitigate the immune response. And that was very informative. Thank you so much. Since we have a paucity of time now, I will just press upon one question which is pondering. Like you have mentioned that the elevated toxicity of Ag+ for cells. Someone has already asked whether you have mentioned some examples of them being used in condoms and others. So what about FDA approval for clinical use of hard nanomaterials like silver? Of course, gold and Fe3O4, you have said, has already been approved. So what are the possibilities of FDA approving the use of these nanomaterials for these, for drug delivery and for these processes?
So regarding silver and regarding, for instance, even copper, etc., and zinc, there is good interest, and there are some trial about external use, especially on silver. In the case, for instance, of condom, is considered an external use. Of course, with injection, I think that the road is much more steeper, but all of this maybe we can reach the clinical approval upon functionalization. So first we need to find safe functional nanomaterials that can be used. For sure, silver nanomaterial, bare, non-functionalized, I don't see them then going on without a surface functionalization.
Thank you so much, Dr. Giacomo Reina. There is one more question that one of our viewers have asked. How have you characterized the BSA conjugated graphenes?
BSA conjugated graphenes, you can characterize them normally via TGA and XPS to do functionalization rate, so to know how much you have it on top. Then you can use even circular dichroism to monitor the presence of alpha helix or beta sheets onto the BSA to see eventually if you have a change in the secondary structure of the protein.
Thank you. With that last question, I thank Dr. Giacomo Reina and Dr. Alberto Bianco for his presence here and to have given us this opportunity to hear from him and learn about his passionate take on H&MS in antiviral research. I'm sure his work has inspired many today, and some of us may even be motivated to learn more about the option of hard nanomaterials as antiviral drugs, and we can even start working on them sooner. Thank you very much. Thank you, sir.
Thank you. Thank you for the invitation.
Thank you. Our last speaker today is the very distinguished and decorated Professor Ayusman Sen.
Should I share my screen?
Yes, sir. Please go ahead. Vernie M. Villaman, Professor of Chemistry, Distinguished Professor of Chemistry and Chemical Engineering, the Pennsylvania State University, USA. In 1970, he completed his BSc honors from the University of Calcutta, India. And then he completed his master's in 1973 from IIT Kanpur. He moved on to the University of Chicago, USA in 1990 and got his PhD in 1978. He is at present, as I said, he's a distinguished professor of chemical engineering at the Pennsylvania State University of USA and has a very decorated career. He received the 2017 Langmuir Lecture Institute of Nanoscience and Technology, India. In 2019, Langmuir Lecture Award, American Chemical Society. In 2019, Humboldt Prize, Alexander von Humboldt Foundation. His research interests are basically focused on synthetic and mechanistic organotransition metal chemistry, homogeneous and heterogeneous catalysis, environmental chemistry, polymer chemistry, energy, and nanotechnology. The topic of Professor Sen's presentation today is enzyme motors and pumps, from transport to collective behavior. I welcome you once again, sir.
Thank you, Devashish, and thank you for inviting me to give this talk. Can you hear me okay? Can you hear me okay?
Yes, you're audible.
Okay. All right. So so this talk is a little bit futuristic, and so it doesn't directly address the current coronavirus pandemic, but I thought that you might like to hear how things are progressing going forward. So the starting point of this research is this little picture on the left, which is actually from a movie. It's called A Fantastic Voyage. And in this movie, what happened was that human beings were shrunk down and put in a nano submarine, and they were able to travel through bloodstreams. So these are red blood cells, these are white blood cells. And so they move through the bloodstream and they repair things in the body. And so this is kind of our inspiration, and that's what we're trying to address. So for some reason, it's not moving. Ah, there it is. So the grand challenge in this area is to design synthetic active matter that interact with themselves, they interact with the environments, and this would lead to interesting collective behavior. And to make these intelligent particles or nanorobots or what you want to call them, you need two things. You need something that can decipher information, and in this case, these are self-powered objects or nanobots. And of course, you need a source of information. And in our case, the source of information is the chemical gradient or an optical gradient. So let me illustrate a simple example. So for example, if you're hungry and you can smell food being cooked in the kitchen, you start walking towards the kitchen. And the reason you know where the kitchen is, even if you cannot see it, is because there's a gradient of odor molecules. So, for example, if fish is being fried, there's a gradient of odor molecules of fish fry. And what you basically do is walk up the gradient. So as you get closer to the kitchen, the concentration of the odor molecules go up, and so you keep going. So that's an example where you are the self-powered object, and the information in this case is the chemical gradient of the odor molecule. And so the combination of these leads to interesting behavior. So I want to talk about this. And before I proceed further, let me just say that you can always move charged objects by applying an electric field. You can move magnetic objects by applying a magnetic field. But that's a lockstep behavior. That's an ensemble behavior. That is not what we're interested in. What we're interested in is to have these nanobots move on their own. So they generate their own power, they harvest the energy locally, they react to the information locally, and they move independently. So it is not any external field that is causing their motion. The motion is being caused by energy harvesting locally, and in our case, this involves catalysis. So what I want to do today is to talk about enzymes for the most part, because enzymes are everywhere in our bodies, and so it's a source, in our case, of power and motion. So one of the first things we discovered was that if you take an enzyme, and actually what I'm telling you is true for any catalytic particle or catalytic molecule, but I'll just focus on enzymes. So what we found is if you take an enzyme and supply it with food or fuel or substrate, whatever you want to call it, and so it starts to catalyze the substrate conversion, it generates enough mechanical force to cause its own movement. So you can see this here. For example, catalase, which converts hydrogen peroxide to water and oxygen. And as you increase the hydrogen peroxide concentration, the diffusion coefficient of the enzymes increases. So power is being generated, impulsive power is being generated, which causes an increase in the diffusion coefficient of the enzyme. If you add an inhibitor, you get the baseline value. So it's clearly tied to the catalytic turnover rate. Now, there have been a lot of speculation in the literature what causes this enhancement in diffusion, but I don't want to spend too much time on it. What I want to move on is to talk about chemotaxis, which to me is much more interesting. By the way, the impulsive force per turnover, if you do a back of the envelope calculation, is about 10 to 15 piconewtons per turnover. And so I want to talk about chemotaxis. So what is chemotaxis? So chemotaxis is the movement in response to a chemical gradient. So for example, when you walk to the kitchen because you smell odor molecules of fish fry from the kitchen, that is an example of chemotaxis. You are chemotaxing in response to the chemical gradient that is the odor molecules of the fish fry. And up until our work, it was thought that chemotaxis was something that only living systems displayed. But what we found is that, in fact, that is not true, and even simple catalytic molecules will chemotax. You can do a number of different kinds of experiments to show this. Here's just one where you have a microfluidic channel with 3 inlets, and you have a uniform concentration of enzymes in all 3 channels. In this case, the enzyme is urease, whose substrate is urea. And what happens is if you pass urea through the middle channel, the urease from the flanking channel will move in and focus in the middle channel. So this is an example of a non-equilibrium behavior because the urease, the concentration was uniform everywhere, but now it's focused in the middle channel. And by the way, you can attach these enzymes to much larger particles, microparticles, and use these enzymes as engines and make these larger microparticles move and chemotax and so on. And we believe that all of this is very important in both biology and outside biology. So I'll give you an example of this. But before we do that, let me briefly talk about the mechanism of chemotaxis. So, if you think about the catalytic reaction of an enzyme, your substrate comes in and it binds to the enzyme, and it can, of course, dissociate again, so it can unbind. And the third thing that can happen is after the substrate is bound, it is catalytically converted to the product, which then leaves. And so there is one way to bind the substrate, and there are 2 ways to have the substrate leave, either by unbinding or through catalysis. And the origin of chemotaxis is thermodynamic, we believe, and that is that if the binding is thermodynamically favorable, the enzyme wants to be where there's a higher concentration of the substrate, and hence it moves towards a higher concentration of the substrate. And you can use a very simple model to describe this. You can talk about a diffusive flow, and it has 2 components. There's a Fickian diffusion, which basically is diffusion of things from higher concentration to lower concentration. And there's a cross diffusion term. And this is the important one, which is the diffusion because of the thermodynamically favored binding. And the cross diffusion term depends on this, which is the Km for the catalyst, the substrate concentration, and the concentration of the enzyme. And if you plug all of this in, you get this. And what you see is that this term, the sign is the opposite of the Fickian diffusion. So where Fickian diffusion moves things from higher to lower concentration, this thing moves in the opposite direction towards higher concentration of the substrate. Now, why, and we've used this in examples. So for example, this is urease again, and chemotaxis of urease in response to its gradient of the substrate urea. So this is the, these are the experimental values, these bar graphs. And the red is the model that I just talked about, and it's more or less fits. It's not perfect, but it does fit well. Now, there's an interesting thing. I said that chemotaxis is because of favorable binding of the substrate to the enzyme. And so just binding itself without catalysis should, in principle, cause chemotaxis. So to test this, we looked at an enzyme which uses two substrates. And so if you give it only one substrate, if that substrate binds and unbinds, there's no overall reaction. And the enzyme in this case is hexokinase, which converts glucose into glucose 6-phosphate using ATP as a phosphorylating agent. And so it has two substrates, glucose and ATP. You need both of them for the catalytic reaction. But if you provide it with only glucose, then the glucose binds and unbinds to hexokinase. And if you give both glucose and ATP, then you get the overall catalysis. And what you see is that with just glucose, you still see chemotaxis. There's no catalysis. But if you provide it with both ATP and glucose, you get a higher degree of chemotaxis and movement. And so the blue is the experimental value, green is the proposed model of ours, and as you can see, there's a reasonable fit between the two. Now, why? So we believe that chemotaxis is actually quite important in biological systems. So let me give you an example. So we have all these catalytic cascades in our body. And so, for example, there's a glycolysis cascade. So the food you eat, glucose, for example, gets converted eventually to ATP. That's the source of ATP in your body. But it does not happen in one catalytic step. It involves something like 20 enzymes. And it's a cascade of enzymes. So the first enzyme reacts with glucose and converts it into something else. The second enzyme then takes the product from the first enzyme and converts it to its own product. And the third enzyme uses that product as a substrate for itself and so on. So this is a handover. And what people have found is that if you provide the first substrate to a catalytic cascade of enzymes, all the enzymes in the cascade will come together. You can actually see them. They're micron-sized dots. You see this in this paper from Science. And then when the substrate is exhausted, the enzymes disperse again. And nature does this for a good reason. And the good reason is that if all the enzymes are together, the first enzyme makes a product, which is a substrate for the second makes a product, which is a substrate for the third, and so on. You just hand it over to the next enzyme and the next enzyme. And so if the product is unstable, you don't lose it. Products don't diffuse into the bulk, so it's much more efficient and so on. So that's all good. The mechanistic question is, what causes these enzymes to come together? And there's been a lot of speculation in biology about why this happens. People have talked about non-covalent interactions between the enzymes, but in many cases, this has never been observed. So our hypothesis was very, very simple. It was that this is all chemotaxis. The first enzyme makes a product, which is a substrate for the second. So the second chemotaxis towards the first, because that's the source of its substrate or food. Second makes a product, which is the substrate for the third. So the third then chemotaxis to the second. So in this way, all the enzymes will come together. So to test this, what we did is, look at the first four enzymes of the glycolysis cascade. So you start with glucose. The first enzyme is hexokinase. The second is phosphoglucose isomerase. The third is phosphofructose kinase. The names are unimportant. The fourth is aldolase. And what we did is we labeled hexokinase and aldolase. And we used this 3-channel microfluidic device again. And in the one flanking channel, we had the first two enzymes, hexokinase and the isomerase. And in the other flanking channel, we had the third and fourth enzyme, the phosphofructokinase and the aldolase. And then you pass D-glucose, which is substrate for the first one through. And hexokinase will immediately chemotax because that's its substrate. The isomerase has to wait until its substrate is produced, then this and then this. So the aldolase has to wait until its substrate is produced by the enzymatic action of hexokinase, isomerase, and the kinase. And so it will have to wait to chemotax a little bit longer because its substrate has to be produced. And so there's a time delay. So the hexokinase chemotaxes quickly, aldolase chemotaxes much more slowly. So this suggests that this is what's happening in your body, the so-called metabolome formation. By the way, if you, instead of D-glucose, if you pass L-glucose, which is not a substrate, then you don't see any of this. And that's expected because there is no chemotaxis, obviously. And so this is an example how you organize active matter, how information can arise from chemical gradients. Because if you have, let's say, 1,000 proteins, and if I give the substrate for one of them, then only those enzymes which are linked together in a cascade will come together. And they will come together in a time-dependent sequential way. Because of what I just told you about the sequential chemotaxis. So this method coming together in a predetermined sequence in a time-dependent way is a good example of how information, in this case, the information is a chemical gradient. And it is maybe relevant to the origin of life scenario. So I'll move on. Let me also say that when enzymes move, of course, they create a disturbance which causes even inert particles to show enhanced diffusion. We've shown this with different enzymes where you can put in tracer particles, inert particles, and they also will show enhanced diffusion. And so there's a couple motion of catalysts and tracers in these systems. And this is actually something that seems to be quite relevant in biological systems. There have now been several papers which have shown that in cells, actual living cells, if you shut off all enzymatic activity, everything stops. And in fact, there's a paper here that I cite where the bacterial cytoplasm goes into a glass-like state, and it's only when you turn on the enzyme that there's a fluidization, and it's because the enzymes start to diffuse and move other things, and that causes all the churning and motion and so on. Let me get back to this chemotaxis thing, and this is, as I said, important in biology. For example, you know that motor proteins move along kinesins. And so this is a cartoon of that. The right-hand side, what you see is a white blood cell chasing after a bacteria. This poor bacteria eventually gets swallowed up. So these are all chemotactic motion in living systems. And so living systems carry out chemotaxis. You do chemotaxis, you go towards the kitchen, there, the bacteria has been ingested. You do chemotaxis when you go towards the kitchen, bacteria will move towards sources of food like glucose, and they will move. So that's positive chemotaxis. But you can also have negative chemotaxis when you move away from something. So, for example, bacteria will move away from toxins and so on. So we've been looking more deeply into this whole phenomena, and so has others. And so you can think about making containers, vesicles, for example, or liposomes, polymerosomes, etc., which you can move using enzymatic reactions. So the enzyme is your engine. You can fill the container with a cargo. This cargo can be drug or some other molecule, and you can cause motion. You can have positive chemotaxis, as been shown by Daniela Wilson. There's an even more interesting one by Battaglia. So it turns out that the glucose level in our brain is higher than the glucose level in our blood. And so what Battaglia did was to have a vesicle, and the enzyme he used was glucose oxidase. And what glucose oxidase does is it converts glucose into products. So the food for glucose oxidase is glucose. So when you have these, you put them in the blood, because the concentration of glucose is high in the brain, they will actually chemotax across the blood-brain barrier. So this is a way of delivering drugs to the brain through the blood-brain barrier. So there have been a number of applications of these things for directed delivery. The previous speaker talked about using nanomaterials for drug delivery. One of the issues with inactive systems is that, for example, if you have an infection in some part of your body, the doctor will administer the drug, but the drug goes everywhere. And so only a very small fraction of the drug ever reaches the infection site. And this is why the doctor has to prescribe drugs at a much higher level than you would normally need. Also, because they reach the infection site by simple diffusion, it takes much longer. So imagine, if you will, that you had these things chemotax specifically in response to the chemical gradient from the infection. Then you will need much less drug because it will be directed and targeted delivery, and it'll be faster because it moves straight to the infection site. So this is kind of the futuristic scenario here. So let me tell you a little bit about, so I've been talking positive chemotaxis. Let me tell you a little bit about negative chemotaxis. And in general, chemotaxis of liposomes. So liposomes are protocells. They are like living cells, except of course, so they have the same kind of membrane that living cells have, but of course they're not living. I won't go through all the details here. All I'll tell you is that we make liposomes. Okay, let me go a little bit here. All I'm going to tell you is that we make liposomes. This is the main lipid that we use, and we attach biotin to a few of them, and we attach dyes to the other ones so that we can visualize them. And we use this to attach enzymes to the walls of the liposome. And you can see this, so the dye has lit up the wall. And if you do fluorescence intensity profile, you can see all the enzymes are on the wall. And these liposomes, just like the single enzymes themselves, will start to diffuse if you provide it with food. In this case, we put ATPase in the liposome wall, and when you provide it with ATP, the diffusion increases, which is this red curve here. And as I told you before, this is directly related to the catalytic turnover rate. So the blue is the catalytic turnover rate, the rate of hydrolysis of ATP to ADP by ATPase, and you can see that they follow each other. So they enzymes, when they're attached to something else, that behave exactly the same way. So the next question was, okay, so the enzymes can power the motion of liposome. The next question is, do they chemotax? And so, by the way, this I don't need to go through this, but you can see if the liposomes are big enough, you can even see them through microscope. But we were interested in much smaller liposomes, vesicles. These are 100 nanometer vesicles, 110 nanometer vesicles. And we can, again, same technique. So these are your lipid molecules. We can attach enzymes to them, these green things. We can attach dyes to them. These are the red things. And then we can add substrate and see whether the liposome moves because of enzymatic activity. So again, it's a 3-channel microfluidic channel, and you have the substrate and buffer through one channel. You pass the liposome and you can have a buffer in the other. So that serves as a control, and you can look at whether the liposome moves towards the substrate or away from the substrate. So towards the substrate will be positive chemotaxis, away from the substrate is negative chemotaxis. So we started looking at this, and so when we put catalase, you see that the liposomes move towards hydrogen peroxide, so it's positive chemotaxis, which is what we expect, because single enzyme molecules also show positive chemotaxis, as I told you before. But then what happens is with urease, I had told you, I showed you earlier that urease, single urease enzyme molecules will move towards urea, positive chemotaxis. But when you attach urease to liposomes, they move away from urea. So it's negative chemotaxis. So this is very interesting. This is the first example of negative chemotaxis. And what's going on here? We were expecting positive chemotaxis based on single enzyme work. And so we started looking into this. And it's somewhat of a long story, but bear with me. So here's some data here. So this is urease-coated liposome, and as you add more urea, it goes further and further away from the source of urea, so negative chemotaxis. So this axis, positive is here, this is negative, so it's moving away. If it's positive, then it's moving towards. And this happens when you have urease present. If you have liposomes that do not have urea on them, you don't see much movement. So what's going on? Well, clearly we were expecting positive chemotaxis. So then we decided that maybe what is happening in this case is the product of urea hydrolysis by urease, which is ammonium bicarbonate, or ammonium carbonate, is causing a negative chemotaxis. So we found out, in fact, that if you take the liposome, you don't even need the urease. If you subject it to the product gradient, ammonium carbonate, it actually moves away. So it's not the urea, but it's the product of the urea hydrolysis that's causing the negative chemotaxis. So we wanted to know, why do you see negative chemotaxis? So I told you what our hypothesis and theory and model is for positive chemotaxis. What's the reason for the negative chemotaxis? So one possibility is the diffusiphoresis. So, if you have ions, ammonium carbonate has ammonium ions and carbonate ions, and they have different diffusion rates. Ammonium ion is smaller and diffuses faster than carbonate ions. And if you have diffusiphoresis, then the motion velocity depends on this, which is the difference in diffusivity between the cation and the anion. Greater the difference, higher is the motion. And of course, since it's proportional to this number, If the cation diffuses more than the anion, then beta is positive. If the cation diffuses more slowly than the anion, then beta is negative. So if you switch the diffusivity, they should move in the opposite direction. And what we find is that no matter whether beta is positive or negative, they move away. So it's clearly not diffusion or transport. Okay, also have osmophoresis because we have more salt concentration one side compared to the other. And so there's a model out in the literature, and we use this model to see if it fits. And the answer is it fits if you put a fudge factor of 10 to the 4th, 10,000, which basically means that it is not osmophoresis. It's not density-driven transport either. I won't go through all this. So what is going on? Well, it turns out that it is an example of what is known in the literature as Hofmeister effect. And this Hofmeister effect is kind of interesting. So you can divide anions and cations according to whether they're cosmotropic or chaotropic. So cations, which are small with high charge, they tend to be well hydrated, and they repel hydrophobic things. Whereas anions that are big and don't have much of a charge are not well hydrated, and they can associate with hydrophobic things. And so people have used these to salt out proteins in these cases and salt in proteins in these cases. And so it depends on the hydration thermodynamics and hydrophobicity, hydrophilicity. And so we believe it's this that's driving this negative chemotaxis, because as you can see, as you go from ammonium carbonate to sodium chloride to perchlorate to thiocyanate, in other words, as you go this way, you go from negative chemotaxis to actually positive chemotaxis. So this is the first example of using this salt effect to cause negative and positive chemotaxis. So this is the story. It's a kind of a long story of how these things move. So, but the bottom line is it means that we can take these vesicles and we can program them and have them move either in the positive direction or the negative direction. And of course, these vesicles can carry cargo like drugs and so on. And so you could, in fact, in principle, use them for drug delivery or other purposes. Let me talk briefly about the third thing. So I showed you first that enzymes, when they turn over substrate, in other words, catalyzed substrate, they show higher diffusion. The second thing I showed you is that when you have enzymes subjected to a substrate gradient, they show chemotaxis. What happens if you prevent the enzymes from moving? Well, if you prevent the enzymes from moving, then the mechanical force is transferred to the surrounding fluid. In other words, they become pumps. And there's no real moving parts in these pumps, but they pump fluid. And the system is very simple. You just take maybe a glass, you put a piece of gold and put enzymes on them. You can attach them by electrostatic attraction or by biotin streptavidin linkages. And then all you do is you put in some fuel or substrate, and you can put in some tracer particles because you need to see how the fluid is moving. You can't see water moving without tracer particles. And depending on the enzyme, the fluid will move in or move out at the bottom. And because it's a closed system, you'll have a convective loop that goes up like this, or you can go up like that. Okay. And so, so here's an example. This is for catalase. You can see the tracer particles moving towards the enzyme. And again, just like the free single enzymes, the pumping rate, and these are in microns per second, increases with increasing reaction rate. So again, it is tied to the reaction rate. Now, of course, it will not go on forever because it's consuming the substrate. So when you run out of substrate, it all stops. And so, but you can resume pumping by adding more substrate. So, for example, if you add more hydrogen peroxide to catalase pump, you get back the pumping rate, or to the urease pump, you get back the pumping rate. So these are interesting pumps. What is the mechanism for their pumping? And we believe that it's a buoyancy-driven mechanism. So if, for example, the reaction that is catalyzing is exothermic and heat is being produced, so this is really a little heater. And so the fluid will move up because the density is lower. It moves up, which draws the fluid from the side. So it is an inward pumping because it moves inward and goes up. On the other hand, if you have a system where the product is more dense than the reactant, then the denser product spreads out this way and more fluid comes in from the top. So this is an outward moving pump. So depending on what's happening, these pumps will pump one way or the other. Now, how do we know that these are density driven? Well, the experiment is very simple. You can take the pump, okay, and if you, for example, if the products are lighter because they're less dense because of heat or whatever, they will rise. So they will go up, and so the fluid will be brought in from the sides. If you now take the same pump and you invert it, and if the products are again lighter or less dense, then they will spread out. They won't go down because gravity, because of gravitational effect. And so now the pumping velocity would remain the same, but they will move in the opposite direction. And that's what we see. That in these pumps, when you turn them over, they move in the opposite direction. And only a density-driven flow will do this. Diffusion or any other thing will cause pumping in the same direction, whether they're upright or not. So that's the mechanism. We were interested in seeing how important thermal effects are. So to do that, we use acid phosphatase, which hydrolyzes phosphates, organophosphates. Here are a number of organophosphates. We know what the velocities of catalytic turnover is. We know what the enthalpy change is, so we can calculate the amount of heat that is generated, because you know the size of the gold patch, you know how much enzyme there is, and so on. And so you can calculate how much heat is being generated. And so our friends at Pittsburgh modeled this. So if you think about it, so this is 1/2 of the pump. And so if it's an in pump, the fluid gets drawn in, moves up, if it's less dense, moves out, and you get this. So this is 1/2 size. So if you look at the velocity, it's in one direction, then it switches, some point it's 0, then it is in the other direction. So this is what the model tells you. And this is what we see by experiment. It looks exactly the same, S-shaped curve. So that looks good, but look at it more closely. And so they did the modeling based on the heat release. And you see that the flow speed is about 10 to the -3 microns per second. And the experimental speeds are in microns per second. So these are about 1,000 times faster. And so you can, as I said, the estimated speeds using just thermal effects is much, much smaller than what we see. And I won't walk you through the whole thing, but the bottom line is that it's the density of the reactant versus product which is causing the motion, not thermal effect. Now, you might ask, so what good are these enzyme pumps? Well, they are actually quite interesting because they're really sensors come pumps, because they will only pump when they recognize their own substrate or some other biomarker. They do not require external power source. You don't need an external power source for pumping. So no mechanical or electrical pumps. And so you can use it for many different applications. For example, you can use it for drug delivery. So one of the things we showed, for example, is you can take a hydrogel, you can fill it up with insulin, and you can put glucose oxidase on the hydrogel. And if you put it in a solution of glucose, glucose oxidase, glucose is your substrate, so it starts pumping, and it can pump out the insulin. Now remember that the pumping rate increases with increasing reaction rate, which means it increases with increasing substrate concentration. And so higher the glucose level in the environment, there's more pumping, more insulin delivered. So it's a drug delivery on demand system. It delivers drug commensurate with how much you need, how much insulin you have out there. So that's one example. We've also used it for antidote delivery. We had a funding from one of the defense agencies who wanted us to look at neutralizing nerve agents. And nerve agents are mostly organophosphates. So we said, okay, give us the money. We'll use phosphatase enzymes because they hydrolyze organophosphate, and then you will neutralize the nerve agent. So they will basically pump in the nerve agent, hydrolyze them, neutralize it, and in the same way as here, they can also deliver an antidote. And it actually works fine. So they have all kinds of mutant enzymes that they make. Here's a special enzyme which will cause this particular nerve agent. This is not particularly so bad. It's a mimic from sarin, but it'll pump. So we said, okay, let's look at a really nasty nerve agent, Soman. So we told them, why don't you attach this enzyme to a surface, add Soman to it and see if it pumps, and you can see it pumps perfectly fine. So in fact, you can use them to neutralize nerve agents and even deliver antidotes. So that's one example of the application. The other is, of course, you can have toxic agents that can inhibit the enzyme. And so this allows you to measure by the flow rate how much of the toxic agent is present. So this is just one example where catalase is inhibited by cyanide. And so if you add more and more cyanide, the pumping speed goes down, and we are able to detect, just by looking at this pumping speed, detect things that are as low as in the parts per billion level, well below the maximum level permitted by our environmental protection agency. So we've looked at cyanide, we've looked at heavy metal ions, and so on. The third thing is sensors. So if you think about sensors, there's of course a lot of work going on in miniaturizing sensors, making them smaller and smaller and smaller. The problem is that if you make a sensor very small, let's say you have a nano sensor, of course, it cannot function unless what it's trying to sense can find it, right? And for something to find the sensor that is nanoscale, it can take hours to days. And this basically shows this. On the other hand, if the sensor is housed within one of these pumps, then the fluid is being actively drawn in and you can have much faster sensing. So for small molecules, it doesn't matter much because they diffuse fast, but if it's a large thing like a virus or a spore, you can speed up the sensing process as much as by 1,000,000 times. So that's another example application of these things. So I guess my time is running out. I was supposed to give a 50-minute talk, I guess. So I think I've shown you that enzyme molecules generate sufficient mechanical force to move things. You can make it directional by imposing a gradient. So this is chemotaxis, and all enzymes show this. And you can make enzymes pumps from it, and we believe that the enzymes provide sufficient force in biology for stochastic motion of cytoplasm, organization of metabolomes, which I showed you, signaling complexes, convective transport of fluids in cells. So I think what we have uncovered is a new area of mechanobiology, which is intrinsic force generation by enzymes and the role in biochemical regulation of cell function. So these are the people who worked on it. These are the talented students and postdocs who have worked on these. Many of them have taken up faculty positions in India. There was Patro is in Mohali in the Institute for Nanoscience and Technology. Krishna is at IIT Gandhinagar, and so on. These are some of the collaborators who have helped us with modeling efforts, both inside Penn State and outside Penn State, and these are people who have funded our research. So this is again our dream. Here's a nanoscale submarine moving through a human capillary, if you will. So what would be the fuel here? Well, there's a lot of glucose in your blood. And so you could use glucose oxidase enzyme as your engine to move these things. So this is, as I said, a motivational dream. Where are we now? Well, this is where the computer mouse was in 1968. This is what it looked like. And now, 40 years later, 50 years later, of course, you know what the mouse looks like. So give us 40 years, 50 years, maybe we'll get to this. So I'll stop here and thank you for listening, and I'm happy to answer questions.
From nanotaxi to nanosubmarine, sir, that was breathtaking. I just don't have words to express the enthusiasm and the interest that you've just created. The greatest challenge of nanotechnology has been the failures in the in vivo applications in biological systems. Your lecture makes it seem all possible now, sir. With such clarity, you have revealed the aspects of attaining chemical control associated with molecular level manipulation of materials, self-propelled mobility resulting from biomimetic catalytic energy harvesting from the local environment, and most significantly, the intelligence and communication capabilities that have been demonstrated in groups of interacting microorganisms. I just am very curious to know, how can nanoscale machines be rendered biodegradable? If so, can they be practically used in the development of nanomedicines without having any side effects or toxicity features?
So that's a great question, and this is the reason why we're looking at enzymes, because these are, of course, catalysts that are present in your body. So by definition, your body makes them and degrades them. We are also looking at vesicles. These are the liposomes, which use these phospholipids which are also the same lipids that your cell uses as membranes. So everything that I've talked about here are biodegradable.
Fair.
And one of the things I should point out is people have started looking at them in vivo. So one example of this is work by Samuel Sanchez, who has used these urease motors for bladder cancer applications. So in bladder, of course, there's a lot of urea. So they move around, and because they generate force, they can penetrate the cancer cells much more than a passive particle can. So they can penetrate deep into these clusters of cancer cells and deliver drugs.
Can specific drugs be tagged to the chemotaxis for subcellular drug delivery?
Well, as I said, you can load up liposomes with drugs inside. And so, when they degrade, or you can pump them out if you wish, that way.
Okay, thank you, sir. The enthusiasm of our participants is well expressed in the questions that are coming in. So I will open the interactive session now and invite questions from the audience. I already have a few. There is one here which says, when it is said the bacterial cytoplasm has glass-like properties, how does it relate to the temperature, as the glass is a supercooled liquid?
So this is actually quite interesting. So I don't think anyone probably understands it, but active particles can change the rheology of fluids. For example, it was shown that if you take bacteria in fluids, and if you align them and have them move, they can drop the viscosity of the fluid in this water by as much as 80%. And then you can come in and you can shine UV light and you kill all the bacteria, and the viscosity goes up to what you expect. So in this case, of course, we don't have bacteria, we have active enzymes. And because they can pump, they can move around, they disturb the fluid, so they're stirring up the fluid, and that's causing a rheological change. So it's not a temperature effect, it is an effect. Think about a little stir bar inside the cell, and they're just moving the fluid around and causing a drop in viscosity. So when you stop the stir bar, everything freezes over. So that's a growing hypothesis. It's not totally understood because no one understands what a nano viscosity is. Viscosity is normally a macroscopic property. So how does a nanoparticle feel viscosity is an open question.
Brilliant, brilliant, Professor Shane. There is another question here, which of course you have already answered. Can this approach be used for the diagnosis and treatment of cancer?
Well, we don't know. As I said, people have been looking at it. They have shown that you can deliver drugs, and you can deliver drugs much more, in a much more directed manner. And that because of the force generated, they can penetrate into clusters of cancer cells much more so. We'll see. As I said, give us 40, 50 years.
Yes, very futuristic. This has been a feast, rather, of futuristic research imaginations towards harvesting dynamic intelligent materials through synthetic chemistry that can counter the constraints of biological systems. All this with the aid of chemistry. I mean, chemotaxis, chemosubmarines crossing the blood-brain barrier could be interpreted as science fiction, but your assurance makes it sound so real. Thank you, Professor Ayusman Sen, for this breathtaking experience of making us aware of what nanochemistry of the future is going to be all about. And I thank you for your time and for sharing your brilliant research experiences with us. I once again thank our speakers of Technical Session 2, Dr. Giacomo Reina, and Professor Ayusman Sen, for making this webinar turn into a festival of the integrated sciences of nanomaterials and their applications in biological systems. Thank you very much. With
Thank you.
this, we come to the end of Technical Session 2, and I now request Dr. Rajiv Panchadai of the Department of Chemistry to render the vote of thanks for the valedictory part of the show. Thank you so much.
Distinguished guest and participant and dear student, a very good evening everybody, and especially good morning to Professor Sen. We are now at the end of international webinar on antiviral drug discovery, creation of armory for the present and future pandemics. In this event, there are more than 1200 registered participants from about 300 academic and research institute of this country under around 30 different disciplines. At the end of the event, it is the time to evaluate the outcome from this conference and to propose the official vote of thanks on behalf of the organizing committee and the Department of Chemistry, Assam University, Silchar. It was a privilege for us to have Professor Somnath Dasgupta, Senior Scientist at IISER Kolkata and ex-Vice Chancellor of Assam University as the guest of honor today. At the same time, very big thanks to all the distinguished invited speakers, Dr. Aviva Levina from the University of Sydney, Australia, Professor Ritu Gaur from South Asian University, New Delhi, Professor Giacomo Reina from Institute of Cellular and Molecular Biology, France, and Professor Ayusman Sen from the Pennsylvania State University, USA, and Professor Alberto Bianco from CNRS, France, who have joined us today in this webinar from different time zone. We are thankful to all of them for their academic contribution, valuable time, and lively interaction throughout the session, covering several issues of current and future pandemics and aspect. The present state of the art of antiviral drug discovery, the role of the soft and hard material, and pointing out the social and moral responsibility toward the survival of humankind. At the home front, I wish to express our thankfulness to Professor Dilip Chandranath, Honorable Vice Chancellor, Assam University, and the Chief Patron of this webinar for his inspiring mentorship. The support of Dean, Einstein School of Physical Science, Professor C.R. Bhattacharjee, who is eventually a faculty member of this department, is thankfully acknowledged. I am also thankful to the Assam University administration in general for providing us with all possible support towards organizing this event. Support from the staff, our research scholar, especially Avijit, Saurabh, Devdulal, Sudeep, Surjatapa, and Amit, and all the students of our department was noteworthy. However, nothing would be possible without the overwhelming response from the participant and their enthusiastic and vibrant interaction that the event was witnessed. Our sincere thanks are due to all of them. I feel sorry to say that there may be certain limitation from our end in organizing this event. I wish you all a successful academic endeavor in the days to come. Hope to get your active support and participation in the future event as well. Wish you a very good health. Stay home, stay safe. Thank you, brother. Thank you all. Over to Dr. Sen Gupta.
Thank you all. Thank you.